Hi friends, my name is Preeti. So till now we have completed autonomic nervous system. So today I'm going to start central nervous system. So before going to start any system, just study the physiology and then pathophysiology and then the drugs, the treatment. Okay, the drugs used to treat that particular disorder and then the mechanism of action of the drugs. Okay, the physiology of the central nervous system, I have done the video on this topic, so I'll give the links in the description. So before going to the CNS disorders, the disorders associated with the central nervous system, first we'll study about the receptors and the neurotransmitters. Okay, neurotransmitters and then receptors which are associated with the central nervous system. So first we'll study about the classification. So this is the classification. There are two types of neurotransmitters which are inhibitory and excitatory. Okay, this inhibitory are GABA and glycine, whereas under excitatory we have fast and slow. Under fast we have acetylcholine and glutamate, and under slow we have dopamine, norepinephrine, and serotonin. Okay, in this classification, the acetylcholine and noradrenaline, these two comes under both the central nervous system and peripheral nervous system. Okay, and acetylcholine and glutamate receptors are ionotropic, whereas dopamine, norepinephrine, and serotonin receptors are G-protein coupled receptors. Okay, these receptors are ionotropic and they are G-protein coupled. Okay, and now we'll go into detail of each neurotransmitter. Okay, first we'll study about the glutamate, which is an excitatory neurotransmitter. Okay, here we can see different types of receptors on which this glutamate acts. Okay, so we call them as glutamate receptors. Under this we have NMDA, AMPA, kainate, and metabotropic receptors. Okay, the first three are ionotropic and the last one is G-protein coupled. Okay, these three are fast acting, because whenever the glutamate acts on these receptors, the response is very fast. Okay, the final result, the cellular effects are very fast. Whereas under G-protein coupled, the response is very slow. So we call them as slow. Okay, because under G-protein, we have a long mechanism. First, the enzyme has to be activated, then there is a production of secondary messengers, then like that, the whole process continues. So it takes a long time to show the response. So it is a slow. Okay. Now here NMDA is N-methyl D-aspartate and AMPA is alpha methyl phenyl acetic acid and kainate. Okay. And this kainate, the exception is this kainate is inhibitory. Okay. But on this, there is an action of glutamate receptors. Okay. We'll study how the kainate is inhibitory. Location where these receptors are located. They are located in cortex, thalamus, and limbic system of brain. Okay. And next, so here we can see. First, this is a neuron. This is a presynaptic neuron, and this is a postsynaptic neuron. Okay, this is an excitatory neuron. Okay. And this is a glial cell. So what happens when a stimulus is, when there is a stimulus, then there is opening of calcium channels, there is entry of calcium into this nerve terminal, and what happens, there is a release of glutamate from the synaptic vesicles into the synapse. Okay, this is the synaptic vesicles in which the neurotransmitters are stored. Okay, here there is a release of glutamate. Now here the glutamate, it can act on the receptors present on the postsynaptic neuron. Okay, or otherwise it can be taken up by the glial cell. Okay, or it can be metabolized. Okay, or it can be reuptaken by the nerve terminal. Okay. So first, whenever it acts on NMDA receptor, okay, this is NMDA and this is the view of this NMDA receptor, N-methyl D-aspartate receptor. Okay, this is the structure of the receptor. So here we can see this is extracellular and intracellular. So on this receptor, there are many binding sites. Okay. First we'll see. So this is the receptor site where NMDA can act or glutamate, the neurotransmitter can act. Okay, this is a major site. And if we come here, this is a modulatory site where glycine can act. It acts as a co-agonist for this receptor. Okay, and this is the channel, ion channel, where the ions will move outside and inside. Okay, calcium and sodium go inside and potassium go outside. And here we can see this is a magnesium. Okay, so now we come to the mechanism, what happens when a glutamate acts on this receptor. Okay, so whenever there is a stimulus, when there is a low stimulus, then whenever glutamate acts, these AMPA channels will open. Under low stimulus, these AMPA channels will open, but under high stimulus only, this NMDA channels will open, because here magnesium is blocking the entry of these ions. Okay, whereas in AMPA it is free. No, nothing is blocking. But in NMDA, this magnesium is blocking this ion channel. So whenever there is a high stimulus, then only this magnesium is pushed out and there is an entry of ions. Okay, here you can see. During the hyperpolarization of the membrane, the magnesium blocks this channel. Whenever that, because if you study action potential, you'll get an idea. Okay, first there will be whenever the stimulus comes, first there will be a resting state. First there will be a resting state, and whenever a stimulus comes, then there will be a depolarization, then repolarization, then hyperpolarization. So whenever this postsynaptic membrane is in hyperpolarization state, then the magnesium will be blocking this ion channel. Okay, but if there is a high stimulus, then what happens? There will be a depolarization of the membrane. Okay, during the depolarization of this postsynaptic membrane, then this magnesium is pushed out and there is entry of calcium. So in NMDA receptor, what happens is the preference is more for calcium ions. Okay, the more calcium ions are sent inside the membrane when compared to the sodium. Okay, so there is an entry of calcium ions and it results in excitation. Okay, so we call it as excitation. And one more thing is, for the opening of this ion channel, it requires both glutamate binding and also glycine also should bind. For opening of this channel, both glutamate and glycine should bind. Then only it opens. Okay. So this is how the response is seen. The excitation is seen. I think you have understood. Okay. We will also see the drugs which act on this particular site. Okay. Now we'll go into the glycine. So glycine is it is an inhibitory neurotransmitter. It is located in spinal cord. Okay, the glycine receptors are ligand-gated chloride channels. Okay, whenever glycine acts on those channels, what happens? There is an increase in chlorine influx, and there will be hyperpolarization, and there will be postsynaptic inhibition. Okay, and here the drugs acting are strychnine, which induces convulsions by blocking the effect of glycine. Okay. See, if the glycine effect is blocked, then what happens? We cannot see the inhibition, there will be excitations. And tetanus toxin, it prevents the release of glycine from the inhibitory interneurons of spinal cord and it leads to hyperexcitability. Okay. So now we'll study about the receptor and the drugs acting on them. Okay, so here we have the receptors NMDA, AMPA, kainate, and glycine. See, all the receptors are named after the substrates which are acting on them. Okay, and methyl D aspartate, it is also one substrate which acts on the receptor. Okay, as it is acting on that receptor, we are naming it as NMDA. And here we can see the agonist and antagonist. NMDA has three binding sites: glutamate binding site, glycine binding site, nothing but a modulatory site, and also the ion channel. And here the agonist is obviously glutamate and antagonist is phosphopentanoic acid. And under glycine binding site, agonist is glycine and the antagonist is kineric acid. See, kineric acid is nothing but kainate. Okay. Uh, that's why we have written initially in the classification it as inhibitory because it is acting as antagonist. It inhibits the binding of glycine. Okay. And ion channel, we have antagonist phencyclidine and also non-competitive antagonist are memantine and ketamine. Okay, this contain magnesium. Okay, and for AMPA, the agonist are glutamate and quisqualate, and antagonist are paracetamol and amlacase. And under kainate, we have glutamate as an agonist. Okay, and then for glycine receptors, we have glycine as the agonist and strychnine as an antagonist. Okay, so these are the drugs acting on the different glutamate and glycine receptors. Okay. So next we'll study about the GABA, dopamine, GABA and dopamine. Okay. Serotonin will study while studying the autocoid, or otherwise I will be telling the serotonin also. Okay. So thank you. We'll meet in our next video.